places still release untreated waste into nearshore areas, right? That they just don't, it just gets pretty diluted, so I think it's okay. So there's some really nasty cases of pollution associated with industry, than people eating the seafood and poisoning themselves in Japan, for example. So it's both. This section we're going to go through basic toxicology, talk about bioassessment. Whoever's been asking that EPT question will bring it back up again. Acid, precipitation, metals, including mercury, organic pollutants, suspended solids, and thermal pollution. So when we talk about some basic terms in toxicology, this is not a toxicology class, but there's some things to think about. One is that an exposure to a toxin can be either acute or chronic, that you can have a big pulse of it, or you can have long-term exposures. And you can imagine that big pulses, you need a bigger concentration to have a bad effect than chronic exposure, right? So a long exposure can build up. It also gets at the idea of whether there's a threshold or not. Is there a level of chronic exposure that you can withstand? And those thresholds below which long-term exposure shouldn't cause any problems are what set the regulatory framework as to what's allowable in things like drinking water. And then you can also think about whether the effects are lethal, sublethal, or cumulative. Do they kill you? Do they make you sick? Or do they make you sick after a long time, after chronic exposure? And to do those, to quantify that, we have the idea of a lethal dose at LD 50, or an effective concentration 50. So lethal, for the lethality, the lethal dose 50. The reason we use a lethal dose 50 is that is the concentration it takes to kill half of the test organism that you're talking about. And the reason for that is that different organisms, let's say we're using rats as our test organism, right? Different rats have different genetics, they vary slightly. What will kill one rat won't necessarily kill the next rat. So you want to find out just sort of on average what it takes to kill them. And that's how the experiments run. Could be with Daphnia, you can't really do that with people, so we have to use model organisms to do that. Likewise, if you're thinking about some kind of effect like lowered reproductive success, an effective concentration that's not going to kill you, but it'll make you sick. So next in the toxic is additive versus multiplicative effects, and this is probably one of the big issues that we really don't know so much about, and that is testing of toxic chemicals generally happens one at a time, but there could be effects of chemicals that are either additive, some are subtractive, so some chemicals would even take away a negative effect of another one, and some are multiplicative, so they're not just straight additive, that they make things a lot worse. And there are so many thousands of organic compounds that we put out in the environment. To understand all their interactions is a very difficult task. There's also effects of other factors on toxins, and you can imagine that any time an organism is stressed in its environment, it's going to be more susceptible to the effects of toxins. So, and you can have interactive things like the effect of UV and acid rain, for example, would interact with each other. Then there's a series of terms about how chemicals move into organisms and then through the food web. And so the first one of these terms is bioconcentration. That is how it's concentrated in the organism. Now it moves from the environment into the organism. It's bioconcentration. The second term is bioaccumulation, and this is the accumulation from its food. And generally this is driven by lipid solubility, because we turn over materials fairly quickly through the liquid process, right? Or most animals do, because they excrete based on the liquids rather than lipids. And it's difficult for them to get rid of lipids. So if something dissolves in lipids, then it's much more difficult to get out of your body. If you have a chronic exposure then to such a chemical, the concentrations will continue to go up and up and up. So that was one of the things that that Rachel Carson put out, is the idea that DDT would build up in the tissues of animals over a course of lifetime, including people, and you'd have these effects down the road that were not predicted. And then the last one is related to organisms in their environment, biomagnification. So if we have a bioconcentration of 10-fold of phytoplankton, then zooplankton eat that, and they accumulate over time. They can maybe be 10,000 times. And then small fish, large fish, and eagles bioaccumulate and biomagnify each level of the food web, brings up that concentration. And so even though we had very low levels of the DDT all the way around in the environment, by the time it got to the top predatory fish, birds, the eagles and such, and then the ones that, the eagles use fish a lot, but the other raptors that use terrestrial animals were building up DDT concentrations high enough that they couldn't form their eggshells anymore. And we almost lost the national bird because of that. So this is just a graph that illustrates the idea of why water solubility and lipid solubility is important on different organic components in fish and mollusks. And so we have things that are very soluble in water, right? They're polar as opposed to non-polar. And they don't concentrate very much. They don't bioconcentrate very much. But things that are very insoluble in water tends to be more lipid soluble, organic soluble, tend to concentrate very heavily. It's a fairly good relationship over several orders of magnitude of both solubility and concentration. The next step is to talk about how we determine toxins in the environment. And bioassessment is one of the most common ways. And this is just using the assemblages that are in the water to determine what the effects are of the pollution. And so we can have ideas as to what taxa are sensitive to what types of pollution. And then, depending upon what type of event we want to see, we use taxa of different lifespans. So you can think of a diatom that would basically maybe have generations in the order of days to weeks, and they would respond, their populations would respond very rapidly to an effect of a toxin in the environment. But if you came back, if there's say some sort of toxic spill that then went out of the system, if you came back in a month, you wouldn't see any indication of that occurring. But if you had sensitive fish in the environment that reproduced maybe once a year or even less frequently, those fish populations, the sensitive fish, would still be gone. So it's a very useful way to basically use the long-term effects of these pollutants to gauge then what you're looking for with the type of organisms you're going to use. The most common organisms used are the macroinvertebrates. And this is for several reasons. One, they're quite common in almost all fresh waters. Two, their lifespans range from maybe months to a year or two at the most. So they don't have huge long legacy effect, but if you can only sample once a month, you're still going to catch an effect, right? Even though you miss the toxin. Three, they're fairly diverse, and so if they have a very diverse group, there's a lot of different functional aspects of that group, a lot of degrees of tolerance, a lot of different food sources that they might be using. Some of them are eating further up on the food web, they're predators, some of them are primary consumers. So all those things mean that macroinvertebrates are very commonly used. Still, fish assemblages, diatom assemblages, a number of different other organisms are used as well, so they're not the only ones that are used. A common index is used in streams as the Ephemeroptera, Plecoptera, Trichoptera, EPT. the proportion that are invertebrates are in those groups, because those three groups tend to be fairly sensitive to most pollutants, and they tend to be indicative of clean waters. So that's a very useful technique. Although it has its problems, it's just one number, right? And it's not based on specific sensitivities. The other alternative is to identify species by species what the species are sensitive to, so you can hone in more on what the negative effect is. So some might be more sensitive to, say, low pH, and others might be more sensitive to high temperature, so you could use those to get at the problems. Related to this, and is commonly used throughout, is the index of biotic integrity, and that assesses habitats and fish species, and it's used throughout the country to get an idea of if a system is healthy or not, if it has what you'd expect, the native of fish assemblage and the native array of habitats that were there. This type of index then has to be calibrated for a particular area, because you can imagine that you'd expect a quite different degree of diversity and habitat types in, say, the Rocky Mountains than you would here. In the Rocky Mountains, the fish assemblage might have 3 species, right? Which, if you came upon a fish assemblage like that in Kansas, that would be a pretty bad system, because we have a lot higher native diversity than we do there. But you wouldn't expect to see warm water tolerant species in Colorado unless there was some thermal pollution in the high areas at least. Whereas here, you'd expect them to be a normal part. So you can see how you have to calibrate these bioassessment tools for the particular area that you're in. In addition, organisms evolve to match their habitat and the range of things that are occurring in their habitat. That's another way of saying this, and those things vary over different parts of the world. Okay, so it is based some on evolutionary responses. So we can think then of some of the different things that organisms might, macrovertebrates might be exposed to. And there's some toxins that the organisms, or some environmental parameters that organisms tend to do best when you're in the middle of a range. And there's the idea of Shelford's law of tolerance, that extremes are bad, but some always have negative effects on diversity. And so we have here the number of species on the y-axis, and then on the x-axis a range of different types of potential environmental stressors. So we see that neutral pH is about, fits this pattern of at sort of intermediate levels having maximum diversity. Very few species are tolerant of low pH. A different group of species, but very few of them are tolerant of high pH. And then you can imagine that if you were making a bioassessment index, you'd figure out which species these were, and if they showed up, you indicates that it's been acidic recently, or at least there was an acidic event in the past, because they're the only ones that made it. Oxygen has the same thing. Very high levels of oxygen are toxic. We haven't talked about that much, but it's actually one of the stressors in eutrophic environments, that the oxygen from photosynthesis gets very high during the day, and then during the night it goes very low. So there's these wide swings in oxygen. And so we see that whether the oxygen is really high or really low can have negative effects as well. In contrast, both chloride and turbidity, basically the more you have, the lower the diversity that you have. And so that's an alternate. Then the question is, when you go into this, what kind of diversity is okay? So if you look at this one, actually, you could say it looks like on average, less than 40 milligrams per liter is probably okay, because you kind of have the same diversity down in here, and then it just falls precipitously, right? This one, if you fit a curve through, maybe, you'd say, well, it's dropping here, but there's this pretty drastic drop right here, suggesting that most of those species over their evolutionary history we're not exposed to that high of levels of chloride and salt. So this is an example of why, when you have roads, streams near roads, that you have low diversity of macroinvertebrates in the streams below that, because in winter they put salt on and the chloride concentrations come up. And then these pulses of chloride that come through, they're not always coming in. It's when that snow melts, right, that it dissolves the salt and pushes it off through the system. So there's this pulse, and you may not catch it, but you could have low diversity because of that if you came back a month later.
Are people trying to use something other than salt now, or are they still using salt?
Yeah, there are alternative forms of salt that are not as toxic as sodium chloride, but they're more expensive as well. So most places what most places do actually is try to use sand and gravel and minimize the amount of salt that they use, and that's the way they deal with it.
Is there any laws saying you can't use certain kinds of salt? Or
Yeah, so different states have laws related to whether you can use salts, and how much you can use. Certainly that you can't just dump salty water into a stream, but when it comes to public safety and roads and whether you treat them with salt or not, then you've got trade-offs, obviously. So now we're going to go through a series of different major issues. Acid precipitation is not one that we have huge problems with here. While the precipitation is somewhat acidic from human activities, we have a lot of limestone in the area, which buffers the effect of that acidity. So essentially, when acid rain falls on our soils, the bicarbonate that's in limestone will convert over to CO2, and the hydrogen ions will be taken up by the remaining hydroxyl ions, just turned into water. So the pH doesn't change very much. That's similar to when you did your alkalinity assay. You basically just titrated a solution with acid until you made the pH change a certain amount, and the more buffering alkalinity there is in there, the more acid it takes to change it. So you can imagine systems that have very low degrees of dissolved salt, such as the glacial lakes, which are basically just scooped out basins of rock, would be very sensitive to additions of acid into their watershed. We'll talk first about sources and geography, and then talk about biological effects. So this is a series of images from the National Atmospheric Deposition Program, which is one that's taken over the last, at least over the last 20 years, taken samples of acid. And we see higher acid being in light green, and low pH, lower acid, high pH, and then low pH being in the reddish brownish colors here. Natural waters run perhaps in the range of, say, around 6. That is because when you take rainwater and evaporate precipitation up, and carbon dioxide dissolves into that water, what immediately forms? Do you remember that one? There's an acid that forms immediately. It's on the test, maybe. Carbonic acid, right? So it causes a slightly acidic pH immediately upon dissolving in there. So rainwater is not exactly neutral, whereas if you just basically evaporate water and measure its pH before it gets any CO2 in it, it's going to be 7 because it'll be pure water. It'll go slightly to the acidic side. But particularly in the upper northeast, there's 2 major sources of acid precipitation, and those are burning coal that has sulfur in it, and that sulfur turns into sulfuric acid in the atmosphere. And there's gasoline that has nitrogen in it, and that nitrogen turns into nitric acid burn fossil fuels. Because of both industry concentrations and high amounts of traffic in this part of the world, the levels of acid are fairly high. The, what one, is it Shell that says they have special gasoline with extra nitrogen in it to make it burn better? That makes me mad. Don't buy their gas. Okay, so here we've got sulfate ion concentrations, and it's just from 85 to 2008, and it's just an animation. We're moving up to 95. We see that as they regulated high sulfur coal and put in limits on the amount of sulfur that could come out of smokestacks, the problem basically got dealt with. And it's just cycling through it, the years down here. And we see that there's a little bit down in here in these high population areas. If you've got coal plants, you've got some coal plants that are burning down in here, and the wind's going that way, so that's making things a little bit acidic. But in the upper Midwest is the one that's the most. So here's an example of something that the regulated community said there's no way we can deal with this, right? It's just, it's too expensive, it's gonna break us. The electricity would be way too expensive for anyone to have. They actually managed to clean it up. So this is a fairly typical course of events when an environmental issue is brought up. So lakes in northern Europe and northeastern United States and southeastern Canada started losing their fish. And they lost their fish because they became more acidic. And you know, it got to the point in Sweden where people with light hair were getting blue hair because the acid was actually eating the copper out of the pipes, and then it would stick on their hair. And so the immediate response was this: how do you know these lakes weren't always acid? It'll cost us too much to fix it, and how do you know these lakes weren't always acid? And so the administration that was in place at that time put money into research. The contrast now is that the people that are in power that are anti-environmentalists realize that they don't want to put money into research because the science will just show that they're wrong, and so they stop doing that. But this is an example of using paleolimnology to figure out what the pH is of a particular lake, and this is a lake in New York. And the cores of the sediments are dated with the lead content because the lead gets deposited, the lead-210 gets deposited, and it slowly breaks down after it's a radioactive compound, slowly breaks down over the years, right? So you get lower and lower content as you go deeper, and that rate is fairly constant. And so from the content, then you can calculate how old the sediment is. So that's how they lock in time here. So time then is given here. And in the 1800s then, at the very beginning of industrialization of the northeastern United States, the pH was about 6. So how did they know that? They go to a bunch of lakes that have a bunch of these chrysophytes that make these scales. They measure the pH, they take a plankton sample, and they just count what species are there. Then they can make a graph that has a relationship between the pH and which species are there. Then, when you come back to the sediments and you count the scales left over from the species, you can say, okay, this is a species here, this is what the pH should be. And we see that, in this case, the pH then, as industrialization happened, in particular starting in 1950, the pH dropped down to about 4.5. Remember, that's a 10 to 100-fold difference in acidities. That's a log scale, right? It's the negative log to the base 10 of the hydrogen ion concentration. And we can also see how some of these species of Malamonas are quite sensitive to acid. So these species were very common early on, and they dropped out of the assemblage, right, as we got more and more acidic. This species. In contrast, some of these species are are basically need acidity to do well, either because they don't compete well with these, or they actually can't do well in more basic conditions. And so we see this one only coming in late on as the acidity came up, this one becoming much, much more abundant. This one is sort of about as abundant throughout. It seems to be a pH insensitive species. Then this pH sensitive species are over here. So this is one of many examples of using paleolimnology to go into long-term effects on the environment and go back in history and see what the chemical changes or the physical changes in the environment were. Well, there's some other chemical effects that then have negative effects. One of them, I mentioned the copper concentrations coming up from the pipe, but also acidic water tends to dissolve most metals at higher concentration. And the bad thing about this is aluminum is quite toxic to fish. It gets in their gills and it rips their gills up and makes it so they can't respire well. And the concentrations of aluminum come up drastically with acid precipitation. So this is just a big data set that the Environmental Protection Agency put together in the northeastern United States and the mountain streams throughout this area. And from fairly neutral waters that have almost no detectable or low aluminum to the most acidic waters down to about 4.5, 5, which have very high concentrations of aluminum. So you can see that changing the pH is then changing the whole chemistry of the system. And then these are just some data to show what happens to the number of fish. And we have here the weekly the aluminum concentration here, and the number of brook trout, and the brook trout just drop off as aluminum concentration comes up. And then in contrast, the acidity is on here, pH, and when it becomes more acid, there's almost no brook trout. So big negative effects, probably mostly mediated by this aluminum. The other thing that happens when you have these animals, such as the trouts, which come out and spawn in the spring, is that the acid falls in precipitation. In the winter, what form is it in the northeastern United States, in the mountains? Snow, good. Snow falls in the mountains, what do you know? Right, so then what do you expect happens in the spring to that snow? It melts, right? So we get this pulse of really acidic situation. This is the time when fish, the salmonids, move into the small stream to reproduce. They put their eggs out. Their eggs are extraordinarily sensitive to acid. They're not protected from the acid like the adult is, right? And so they're putting their eggs out at just the time when this pulse of acid comes down through the system.